Mutational burden in male cancers is higher than the mutational burden in female cancers. This presumably reflects the overall trend for higher mutational burden in males. Interesting to note that following Hiroshima and Nagasaki, there's higher rates of cancer in male survivors as compared to female survivors. Males with inherited DNA mismatch repair defects, as in Lynch syndrome, have higher risks of colorectal and other cancers than females similarly affected. It's important to know that both estrogen and testosterone do regulate DNA repair, but they don't do it identically. And the response to DNA damage can be quite different in male and female cells, as we've been studying in my lab for several years now. As an example, in response to DNA damage, like irradiation damage, female cells will undergo senescence more readily than male cells. So among the things that may contribute to the sex difference in incidence is the sex differences in mutational burden and sex differences in DNA repair. Now, when DNA is damaged, among the ways that cells intrinsically protect us from cancer are the numbers of tumor suppressor pathways and networks. Senescence is one of those tumor suppressor mechanisms, and so we already have some evidence that maybe tumor suppressor mechanisms may be different in males and females. Pictured here on the left is a finding from a study from Chen and Arnold regarding this is actually from a later study from Haupt, a finding related to investigating why male and female mice that are null for P53 have different neural tube defects. So when you simply knock out P53 in mice, what happens is more male pups are born than expected and fewer female pups are born than expected. The ratio is off. And that's because the females die in utero. And the reason the females die in utero is they have anterior neural tube defects and they die. It's incompatible with life. The males have neural tube defects, but they are posterior caudal neural tube defects, and they are born basically with spina bifida. That difference in loss of P53 effect is consistent with a number of bits of data in developmental models and in aging models that suggest that male and female utilization of P53 as a regulatory mechanism and as a tumor suppressor may be different. It turns out that this is because of the way that P53 interacts with the X chromosome. There are a number of alleles on the X chromosome that escape X inactivation, and they are modulators of the P53 pathway. There's a beautiful paper from Dunford et al. From several years ago about this group of genes. Among the downstream mediators of P53 effect is the gene P21, which is expressed at higher levels in female cells across multiple species, as described again beautifully by Navki in Science just a few years back. And we most recently have published a paper that I meant to include on here about sex and mutation-specific gain of function in P53 activity. This tumor suppressor pathway, which is at the core of all tumor suppressor activity, functions differently in male and female cells, and presumably that also contributes to the cancer risk that we see. And if we had time and space, we would also be talking right now about anti-tumor immunity, but in the interest of time, we won't, other than to say that anti-tumor surveillance function of the immune system is quite different, and there is very good evidence that it's more effective in females than it is in males. And there's good evidence that that relates to the significant sex differences in response to immunotherapy for cancer. Males respond better to immune checkpoint inhibition than females do. Presumably among the reasons is that the female immune response is already maximized, whereas the males are not. So that probably contributes to the difference in sex, the sex difference in cancer incidence as well. I want to switch to thinking about survival. And I want to remind you that survival is really a measure of response to treatment. And treatment, radiation, chemotherapy, and surgery can be quite different based on some pretty fundamental differences in pharmacokinetics and some differences in pharmacodynamics. So for anybody interested in the sex differences in pharmacokinetics, you must read this pharmacological reviews that Frank piloted from just last year. It's a fantastic and comprehensive analysis of fundamental differences in pharmacokinetics and pharmacodynamics in males and females. There's evidence for difference in drug absorption. Here on the upper left, I'm showing you a profile for the absorption of fluconazole after an oral dose. In addition to oral absorption being different for many drugs, pulmonary absorption, transdermal absorption are also different. So same dose of the same drug in a male and female may result in different plasma levels. If we look at liver metabolism, and this is just a small subset of liver metabolic genes, they differ in activity between males and females. And so any drug that requires liver action for activation is going to be activated differently, either total magnitude of activation or the kinetics of activation in males or females. Drugs that are inactivated by liver metabolism will also have different activity. Volumes of distribution are different between males and females. Lipid-soluble drugs have a greater volume of different distribution in females, while water-soluble drugs have a greater volume of distribution in males. Again, these would suggest that maybe we should be dosing these things with greater attention to who it is we're treating, rather than just their weight or their body surface area. And elimination can be quite different. So here is a paper from quite a few years ago about childhood leukemia treatment, which includes methotrexate and 6-mercaptopurine. If you just look at the MTX rows here, you can see that the elimination, this number 4, is statistically longer than in girls than it is in boys. Number 3, since methotrexate exposure area under the curve determines both efficacy and toxicity, this is actually quite a big difference. And again, we don't routinely dose these things differently. As an example of how important this can be, we can look at the drug 5-fluorouracil. This drug is very frequently used in adult cancers. As far back as 1992, it was demonstrated that females exhibit slower clearance of 5-FU. In multiple clinical trials involving thousands of patients at this point, females had more 5-FU attributable adverse events. Adverse events, toxicities, are a concentration of exposure, dose, and time. And so the slower clearance of 5-FU would be anticipated to have a greater toxic effect in females than it does in males, and that is borne out by the data. As a consequence of that greater toxicity, females complete fewer planned chemotherapy cycles than males. This is an important issue when we're thinking about what the required total doses of drugs are to cure a cancer. But rather than these fewer planned chemotherapy cycles being associated with decreased survival in females, females actually have longer survival than males do when they're treated with 5-FU. And presumably this is reflecting the same process as the toxicity data. Their cancer is exposed to more of the 5-FU at the same time that the normal tissues are. Similar data, and there's just the male-female difference for the survival. Similar survival advantage with 5-FU for females has also been been reported for esophageal cancer, in addition to the colorectal cancer I forgot to mention above. So the pharmacokinetics are significantly different routinely between males and females. It matters for drug dosing, including chemotherapy, yet we don't really do enough to take that into account when we're taking care of patients. Survival is also dependent upon pharmacodynamics. This is particularly true in the era of targeted therapeutics. Single mutations in single targetable genes and proteins may have very different roles in male and female cancer, and there's multiple reports to this effect at this point. These are three more than three panels from a paper of ours from a couple of years back, where we were looking directly at the response of male and female glioblastoma patients to radiation and temozolomide chemotherapy. In collaboration with Kristen Swanson, an imaging scientist at Mayo Clinic, we were looking at the MRIs for patients at the time that they started their first cycle of temozolomide post-irradiation, their second and third, actually first two cycles, second, third, and fourth cycles, third, fifth and sixth cycles. And from the MRs, we could calculate a growth velocity for the tumors using an algorithm that Kristen developed. And what you can see on the left is that the female growth velocity declines with each successive cycle of temozolomide. The males decline initially, but then they plateau. And in fact, as we've said, the females survive longer than the males do for GBM, as they do for so many other cancers. And we were able to see that, and we were able further to correlate these survival data with gene expression data and extract from the male and female gene expression data, male and female gene expression signatures that correlated with response to radiation and temozolomide, or survival. And those signatures were completely different in males and females. The genes that were most highly correlated with survival in females, so the longest surviving females were distinguished from all the other females and from all the males by decreased expression of everything that's in blue here that we believe is functioning within an integrin signaling pathway. No time to talk in detail about that today. And these three genes that also function in this pathway and were upregulated in females. For the males, survival, again, those males that survived longer than any other males and survive, well, not longer than the females, but survived longer than any other males, included downregulation of all these genes associated with cell cycle regulation and upregulation of these two. So were you to target something in these pathways, let's say we were going to target EZH2, the methyltransferase responsible for trimethylating H3K27. Let's say we were going to target that. We may have some effect in males, but it's unlikely that we would have much of an effect in females, even if the level of expression was the same. And we showed that in this paper, that it's not the level of expression that determines it, it's how the gene and protein are utilized. And others have shown the same, most notably Camila Lopez Ramos has shown beautifully that transcriptional networks that are regulating metabolism can have the exact same levels of expression in male and female cells, but be associated with very different levels of activity. So the pharmacodynamics, the value of inhibiting a specific target based on that target's activity can differ quite a bit in males and females. And this, of course, can also contribute to the difference in survival. So these differences in incidence and survival are related to some pretty fundamental aspects of male and female biology. And this should come as no surprise to us that they have an impact on cancer. And so first, we're going to talk a minute about development and sexual differentiation, because sex differences in biology are evident from the moment of fertilization, from the very first hours of after fertilization. There are differences in the expression of DNA methyltransferases. Male DNA tends to be hypomethylated compared to female DNA. And there are differences in metabolism. Male pre-implantation blastocysts metabolize more glucose than female pre-implantation blastocysts. They generate more lactate. The females, of course, have higher pentose phosphate shunt activity because it's dependent on G6PD, which is an X allele that escapes exon activation. But by day 2, male embryos across species are larger and contain more cells than female pre-implantation blastocysts. And of course, those growth differences persist actually throughout life, as we'll talk about in just a second. Across all the stages of life, there are different eras in which different events are occurring in sexual differentiation. Pre-implantation, there's sex-specific reprogramming of imprinted alleles to favor greater growth rates in male versus female pre-implantation blastocysts. Much of that is the result of the difference in sex chromosome complement. Exon activation equalizes dosage for many genes, but not all genes, including a network of tumor suppressors and growth regulatory genes. Epigenetics differ from the moment of fertilization, both histone and DNA methylation, and that persists throughout life. And metabolism, as we've just said, differs from the moment of fertilization. During in utero development, there's a period of time when there's a surge in primarily testosterone, but also estrogen, which patterns gene expression for life. It patterns tissue structure and gene expression for life. There's a short, less high amplitude surge in circulating sex hormones just around the time of birth. But then through the pediatric years, there's no circulating sex hormones until puberty. And though it's drawn here as if estrogen is constant, estrogen's in the red line throughout adult years, obviously it's not, it cycles. Testosterone is constant, and then in the menopause years, circulating sex hormones decline. And so biology can be determined by the sex chromosome complement. It can be determined by the epigenetics, and it can be determined by the epigenetic effects of in utero sex hormones, epigenetic effects of perinatal sex hormones, and then the combined epigenetic and acute actions of circulating sex hormones later in life. This also shouldn't surprise us because these sex differences have been evolutionarily selected for to optimize reproductive success within a species. And the species reproductive success is dependent upon different biologies in males and females. We can broadly refer to females as the big gamete producers and males as the little gamete producers. There's a useful bit of math that's helpful in understanding the evolutionary process, which is the per gamete or progeny resource sacrifice. So for the large gamete producers like females, they make all of the ova they will ever have prior to birth. 1 to a few become available for gestation at any given time. Pregnant females provide all of the personal resources for the gestation of fetuses, the nurturing of newborns. And in fact, in long-lived species, females need to survive into grandmother years for fully optimized reproductive success. And so they are said to be resource limited in their reproductive success. And evolution has worked to enhance those resource allocation control mechanisms in order to balance the resource needs of mother versus progeny during gestation and during the newborn years, and then to preserve longevity, which requires tremendous resource allocation with regard to tissue maintenance, a fantastic subject to think about with regard to regenerative medicine. Little gamete producers, on the other hand, produce enormous numbers of sperm throughout their lives. Males are physically capable of impregnating multiple females every single day. They provide no personal resources for the gestation of fetuses, nurturing of newborns, and their survival into grandfather years is not required for success of the species. They are said to be ova or mate limited. Consequently, evolution has worked to make them successful, basically in competition for female mate choice. And so males are highly ornamented and they're colorful and they engage in competitive behaviors with other males that are meant to influence female mate choice. These are two very different biologies, two very different strategies for reproductive success. And consequently, evolution has worked differently in males and females across all sexually reproducing species. And as a consequence, again, across species, there's sex-biased actions in growth. We use two different growth charts for kids even before puberty, because even though there's no circulating sex hormones, they are programmed to grow differently. Males faster than females. Females then have a pubertal growth spurt that happens before males, but then they plateau at a height during their teen years that is lower than the male plateau, and males continue growing into their 20s, actually. And interestingly, body size is correlated with cancer risk in humans. On the top here, you'll notice a trend that goes from the bottom left-hand corner to the upper right-hand corner. This is the hazard ratio for cancer as a function of height. The top graph is in women, where you can see the taller the women are, the higher their cancer risk. The bottom graph is for men, where the effective height is a little bit narrower than it is for females, but it's still present there. So growth is a risk factor for cancer, and growth is fundamentally different between males and females. So is metabolism, as we mentioned. And on the left here are differences in serum metabolites in healthy adult male and females. And you can see everything to the right of the dotted line is more abundant in male serum. Everything to the left is more abundant in female serum. In red boxes are the super pathways, which are indicated on the left, amino acid, carbohydrate, and then in blue are sub pathways. If we think just about carbohydrates for a second, in which the metabolites are more abundant in males, reflecting their greater utilization of glucose, which again was true from the moment of fertilization. We can look at cancer risks in males and females as a function of per capita glucose consumption. And what you can see in the triangles here is that male cancer incidence goes up more steeply and to a higher level as a function of increasing sugar intake than does the female rate of cancer. So again, fundamental sex differences affecting a fundamental process like metabolism, which impacts on cancer incidence. And then finally, longevity. As people live longer, the sex difference in longevity in humans becomes more and more apparent. The blue curves are after 1950, the brown curves are before 1950. And this is true across species. I've pictured squirrels here because there's such a stark difference in the lifespan of male and female squirrels. And of course, cancer is really a disease of aging more than anything. And in humans, you can see that with age, the rate of cancer goes up in both males and females, but it goes up more steeply in males than it does in females. So again, sex differences, fundamental ones, are affecting fundamental processes that are impacting on cancer incidence, such as growth, metabolism, longevity, DNA repair, metabolism also having an effect on mutational burden. And each of these things also, these pathways plays a role in response to treatment. And so we can conclude from this portion that, of course, there are sex differences in cancer that impact on cases and survival. We talked about a few of these, but not all of them. I would refer you to, if you're interested, to a review that have just had published in Trends in Cancer. I should have put the reference up, but it's just brand new. And the takeaway from this is, one, it shouldn't surprise you that there are substantial sex differences in biology under normal circumstances, but also in disease. And that all drug development, all preclinical studies, all clinical trials should be powered to detect those sex differences in efficacy and toxicity if we want to do the best for our patients with regard to curing them and doing it with the least toxicity. So now we're going to switch gears here and talk in more depth about sex differences in GBM glutamine metabolism. And then we're going to ask this important question about what would sex adaptation of treatment look like. I realize I took a little bit longer than I intended, so I might talk a little bit quickly. So sex differences in metabolism have been known for 100 years. First described for sex differences in cholesterol levels. Then in the 80s was all that blastocyst work, which was actually done by the meat industry first because they use in vitro fertilization, and they discovered that they needed different normative values for male and female blastocysts to screen for those that were most viable. And then in the 2000s and continuing into now are the very substantial differences in adult nutrient utilization. Those differences in metabolism exist at the cellular level, the tissue level, and the whole organism level. They're present from the moment of fertilization through childhood and adulthood. Metabolism is a critically important thing to think about when you're thinking about sex differences in biology. Yasmin Spinagel, who just defended her PhD, is the person who did all the metabolism work I'm about to describe. This aspect of the work was also done in collaboration with Joe Ippolito and Prakash Chinayan. So here is metabolomics from, I don't know, close to 70 glioma specimens from males and females. On the top, you can see who's male in blue and who's female in red. We have ordered this by metabolite abundance. So on the left is the most enriched samples for metabolites. On the right are the least enriched. And I think you can see that on the left is purely males, and on the right is mostly females. And then there's an area of overlap in the middle, and this will be very important. This concept of males and females at two different ends and a lot of overlap in the middle will be important as we move forward. The super pathways are the same as they are in the normal serum, and that's important. Cancer cell metabolism really is a reflection in many ways of normal cell metabolism when we're talking about sex differences. So among the metabolites that's very different are components of the glutamine pathway, and this is very important because glutamine metabolism is being targeted in clinical trial right now. And so here, in collaboration with folks at Sloan Kettering, we looked at male and female glioma patients with standard MR and glutamine PET. And on the bottom row is the fused image. So here we have two cortical GBMs that are similar in size and similar in gadolinium enhancement. When we look at their glutamine uptake by PET, we see that the male tumor is uptaking much more glutamine than the female tumor is. We can quantify that, which is quantified here in the graph on the left, and we can compare it to the normal brain. And again, there's still a sex difference in the normal brain because these sex differences exist fundamentally, but it is actually greater in magnitude in the tumor than it is in the normal brain. We can look at this in one of our mouse models, and the sort of workhorse mouse model we use is one in which we have deleted neurofibromin and abrogated p53 function with a dominant negative. We have other models, but this has been the model we've used for years now. And we can see that when we look at this model, that the glutamine consumption reflects what we saw in the human patients. Males are consuming more glutamine than females are. And if we looked at the sensitivity to glutamine deprivation in male and female cells, we see that the males have a left shifted dose response curve compared to the females. They are more sensitive to glutamine deprivation than females are. So why might this be so? Well, glutamine is utilized in many different pathways. So here in the center on the top is glutamine coming into the cell. It's acted upon by glutaminase to make glutamate. The glutamate can go into glutathione synthesis, or the glutamate can go to alpha-ketoglutarate amino acid synthesis via transamination. Alpha-ketoglutarate can then go directly to citrate, and fatty acid synthesis can go into the TCA cycle, where in addition to replenishing the TCA cycle and generating energy, it can be it's from the citrate that it gets into the fatty acids. So we looked at all of these things to see where the nitrogens and the carbons from glutamine were going. We had nitrogens from glutamine going into nucleotide synthesis, carbons going into lipid synthesis, but there was no significant difference in those. Where we saw significant differences is in glutamate being incorporated into glutathione, which is here in C. Small differences, but significant. The biggest differences were in glutamine-derived carbons going into other amino acids, and you'll note the branch chain amino acids in particular. Were very differently regulated in male and female cells, and other TCA cycle intermediates were as well. There's greater flux through the TCA cycle in males versus females. That's true overall. Again, it reflects the decreased efficiency, so there needs to be greater flux. But the way that the carbons and nitrogens, well, the carbons from glutamine are partitioned is different in males and females. And glutamate is key to this. And so we look specifically at glutamate expression in GBM patients. There are two isoforms of glutaminase, GLS1 and GLS2. You can see that GLS1 is more highly expressed in both male and female GBM specimens compared to GLS2, and that in vitro in patient-derived GBM cell lines, it's also true that GLS1 expression is higher in males. In the glioma cell lines, we can see that GLS1 expression is actually higher than it is in females. We saw that in the patients, but of course it's harder on the with all the different data points here. GLS1 has two different isoforms, KGA and GAC. We looked in our mouse model at which isoform was most highly expressed, and it turns out that it's the GAC isoform that's most highly expressed. And so we looked at glutaminase dependency, because glutaminase is the target for drugs that are currently in clinical trial. And what we saw is if we use the lead compound for clinical application, CB839, that just like the glutamine deprivation, males exhibit a significantly left shifted dose response curve. They are very sensitive to the inhibition of glutaminase, just the same way they were very sensitive to glutamine deprivation as compared to the females. And that was also true for a second inhibitor, BEPTIS, which you can see there. Why is this so? And I'm just condensing an enormous amount of data here. We did a series of rescue experiments to try and determine what it was that was so critically important about glutamine to glutamate for males versus females. And it turns out it's because this is how the males are replenishing their TCA cycle almost exclusively. The way we did this experiment is we used a membrane permeable form of alpha-ketoglutarate. You'll recall that glutamine goes to glutamate through GLS action, and glutamate can go to alpha-ketoglutarate and enter the TCA cycle. So in this case, under CB839 conditions, we can see the relative cell numbers of male and female. Males are much more sensitive than the females are. When we add dimethyl alpha ketoglutarate, we completely rescue the phenotype for the males. There was no phenotype to rescue for the females, and so we don't see any change. We can contrast that to an effort at pyruvate rescue. Pyruvate can replenish the TCA cycle in two different ways. It can be directly enter the cycle, or it can be converted by pyruvate carboxylase into oxaloacetate. So here we find that pyruvate can rescue the male phenotype slightly, and it has no effect on the females. And so we looked at this pyruvate carboxylase to determine whether or not the males are able to use pyruvate through pyruvate carboxylase to replenish the TCA cycle. And it turns out that males express very, very little pyruvate carboxylase. As you can see, 3 biological replicates up here, normalized Western blot data here. And we can further test this by looking at the incorporation of pyruvate carbons into intermediates in the TCA cycle. When the pyruvate comes into the TCA cycle through pyruvate dehydrogenase and acetyl-CoA, there's only 2 carbons from pyruvate that make it their way into the intermediates. When pyruvate is converted by pyruvate carboxylase into oxaloacetate, 3 carbons are incorporated. And so we can look at the M+2, this is the 2 carbon isotopologues, and we can look at the M+3 isotopologues after administering carbon labeled glutamine. As you can see, the M+2 route, pyruvate through acetyl-CoA, males exhibit more of this for most of the TCA cycle components, greater flux in males than females. But if we look at the M+3 isotopologues, you can see that the females have much more than the males do. And so, so we conclude from this that male and female glutamine metabolism, which again is actively being targeted in clinical trial, fundamentally differs. The amount of glutamine required for male and female cell viability is different, and that among the reasons it's different is that males require it for replenishment of their TCA cycle, and they have higher levels of TCA cycle flux, so greater requirement for glutamine. Females, on the other hand, can replenish their TCA cycle with glutamine through alpha ketoglutarate, but also through glucose to pyruvate to oxaloacetate, something that the males don't seem to be able to do as well. So it would seem that if we want to target glutamine metabolism in clinical trial, that we would have to adapt our treatments for males and females. And, you know, that's all well and good, but there is a big problem here. And the big problem is illustrated, and I'm going to try and be brief here to just cover a small bit of this. I know I'm late. This is an adult female anglerfish. Attached to her perineum are 2 adult male anglerfish. When these adult male anglerfish got to sexual maturity, they digested their entire alimentary canal, found the perineum of a female, attached themselves to her, digested away her skin, secreted angiogenic factors so that their circulatory systems would become one. When their circulatory systems were unified, the males went ahead and digested other organs like their liver and their kidney because they just weren't going to use them anymore. And they became nothing more than testicles for the female. This is sexual dimorphism. This would be, you know, you could think about male specific versus female specific here with no problem. Humans aren't like that. Sex differences in humans are more commonly differences rather than dichotomies. Obviously, testes versus ovaries is a dichotomous difference. But most differences are like height. This is an old picture. In white are the female college-age students, in black are the male college-age students. What you can see is that the shortest end of normal height is exclusively females, and at the tallest end of normal height is exclusively males, but in the middle are numbers of overlapping individuals. This is like that metabolite heat map I showed you, where at the extremes, it's really pretty much male versus female, but most of the samples were in the middle. So if that's the case, how do we take sex differences and turn them into treatment modifications? It cannot be that we're going to have male specific and female specific treatments. We're going to have treatments that incorporate a spectrum of sex differences, treatments that will place individuals wherever they are in that spectrum, from the extreme male to the extreme female, and do so in a way that generates insight into what the mechanisms are, what the PD would be that we could target in both. And I recognize that there's less than 10 minutes left, so I want to skim through something to show you how we're doing this, because I think this is going to be critically important to how we move this field forward to clinical application. There's a couple of key concepts. One is about bias in action. So if we think about growth, there's a female growth optimum and a male growth optimum. And there are biased actions, male versus female, that include the genetics, the epigenetics, and the acute hormone actions of sexual differentiation. And that for individuals that are not at the optimums, for males that move into this mid-range, the overlap range, you can think about them having either increased female biased actions or decreased male biased actions, and the opposite for females. And if we think about multiple axes rather than height, things like regulation of cell division, response to DNA damage, response to glutamine deprivation, or some of the epigenetic mechanisms, you can start to see how we might apply the concept of a spectrum of sex differences to adaptation of therapy. And the way that we've done this is kind of illustrated here. If we thought about height and we thought about its relationship to proliferation, cellular proliferation, and we wanted everybody to be at the female end of this spectrum, because that's where the lowest, for cancer treatment, because that's where the lowest proliferation was, we would think about how it might be different to push a female patient, let's say one in the mid-range, towards the female optimum, as compared to taking a male patient in the mid-range and pushing them towards the female optimum. And I'm going to illustrate this because I'm just going to rush now. We did this with a Bayesian nearest neighbor analysis. So I'm going to take you through it rather than talking about these details. So here's an example. This is kidney clear cell carcinoma, male-female incidence ratio of 2:1, and females have a survival advantage. Common theme in cancer. We can take the entire transcriptomes of this few hundred patients here, or maybe 100 patients here, and we can use a Bayesian nearest neighbor approach to determine their similarity to each other. And when we do that, we can find areas that are exclusively female and areas that are exclusively male, and we can assign them a value that will translate into something similar to the tallest and shortest height, if you will. And then we can look at the distribution of the population for those values, and you see something very similar to what we see with height. It's male distribution, there's female distribution. At either end is exclusively males or females, but most of the individuals overlap in the middle. And we can use that to start identifying what the pathways are that are exclusively male and exclusively female. You can see some of them illustrated here. And we can use that across cancer types. Again, I'm not going to go into detail, but you can see one of the major polarizations in cancer across 26 cancer types is that female cancers at the pole, the optimum, are really all about immunity, and male cancers are really all about cell cycle regulation. And we can further identify what the differences are for male or female individuals that are in that overlapping mid-range as compared to their poles. And the value of this would be, as we think about individual patients using their whole transcriptomes, placing them along an x-axis of any of those things that we talked about before. It could be proliferation phenotype, metabolism phenotype, immunity phenotype. And if we wanted to target those phenotypes with like a CDK inhibitor for the cell cycle analysis, we would have greater insight into how to do that for males versus females by understanding what the pathways were that were activated to bring them from their optimum, so female from this end into the middle, and a male from this end into the middle. I know that probably doesn't make a huge amount of sense because I had to rush through this. It's an exciting concept, and it's one that we're really trying to push forward, and I apologize for having too much other stuff. But I'm going to end there. Sex differences in cancer mechanisms are substantial. Adapting cancer treatments to patient's sex is going to need to account for a spectrum of sex differences. It's not going to be dichotomous, and that will complicate everything, but it's not insurmountable. I want to thank all the amazing people in my lab and all the collaborators and all my funders. And I'm going to stop sharing and stop there.